if I'm going to convert it into, what's it called? Um, into PowerPoint formats, I think you might not really, I don't know how it's going to work. If a lot of you guys will be able to understand most of the information. And since you have the textbook, which is formula biology, I know a lot of you will have it. So since you have the textbook, it's just like me carrying the textbook and moving it into PowerPoint. So it will be in form of a presentation. So that will make it very easy for you to go through your whole textbook when you are reading it. And I try as much as possible not to remove anything from it, although I did some little adjustment and other stuff. But basically, if you still carry the textbook and you begin to go through it one after the other, you will understand everything. So another way of reading all this is you carrying the textbook, and as I'm teaching you, you are jotting inside it. It's your textbook, so you can do anything you want to do into it inside it. So you jot it, you take note of some important information, and that will make it very easy. And all the part of questions and everything that are there, I might not put all the questions here. So in which I expect you, since you have the textbook, go to the questions. Since we have done at least one example in this whole explanation, at least one example of every question IJMB and JPEB has ever asked. So we've solved it, one question at least from this what's it called, these classes. Now we want to go to dihybrid inheritance. And is it dihybrid inheritance? I'm coming, please. Okay. Okay, yeah. It's going to be monohybrid inheritance. I think the last class we were able to talk about the four what's it called? The 46 terms used in genetics. So now it's more like we are trying to expand on it. We're trying to see more on it, which for those of you that have been going with me, that has been following me, you would notice most of those things I'll be mentioning here, most of them has been mentioned while I was explaining each of them. Monohybrid inheritance, I've talked about it before. Mono means one, hybrid. So hybrid is definitely gotten from species of unlike parents. Yeah, species from unlike parents, and inheritance. So inheritance of one character from unlike parents. Now, what do we mean by unlike parents? It means, okay, for example, now we have a parent, a parent plant that is giving rise to a white flower. We have another parent that is giving rise to a red flower. So they are unlike. So we now cross them and see what is going to happen. So like I told you, Gregor Mendel is actually an Australian monk, and he worked on a particular plant which is called a garden pea. That is Pisum sativum. And monohybrid inheritance actually, as I've earlier told you, inheritance of one pair of contrasting character. I told you in every organism there are a lot of characteristics. So in the plant that Mendel actually worked on, which is this garden pea, or we call it our pea plant, this very plant has 7 different characteristics. But out of all the 7 different characteristics, in monohybrid inheritance, he only picked one and talk about that one. Now, let's look at the 7 characteristics that that the garden pea, which Gregor Mendel studied. Let's see the 7 characteristics that we have in this very plant. We have length of stem, which means some stems are tall, some are short. We have shape of seed. Some seed are round, some are wrinkled. We have color of seed. Some seeds are yellow, some other seeds are green. We have color of unripe pod. Some are green, some are yellow. We have color of flower. Some are red, some are white. So that is color. Position of flower. Where are the flower? At the axial part or at the terminal part? Shape of the pod. Inflated or constricted. What are we trying to say? Now, let me bring it to the human kingdom now. Now, we have a lot of characteristics in human too. The type of nose, the shape of your nose, the type of eyeball you have, the type of hair color, your type of mouth, I know you would have noticed that there are different type of mouth, a mouth shape rather. The type of ear, the type of a lot of things, a lot of characteristics. So all these are characters or characteristics which are found in an organism. So monohybrid now, out of all these 7 characteristics, so if you want to talk about monohybrid inheritance, we'll just pick one and talk about that only one in 2 different species, which are called hybrid. Now, everything we have here is more like what I explained earlier, but all the same, I just want to go through it. So, I told you that is a monk. So, he studied only one or at most 2 characteristics at a time. So only one character or 2 characteristics at a time. That's what he studied. And he studied easily contrasting character. Easily contrasting character. This one is red, this one is white, this one is short, this one is tall, this one is this one is that. Easily contrasting, so you can easily see it. And the very good example is the red, that's the color of flower. Red flower, white flower. So from there, he went through the first filial generation, which is F1 generation, to know what is going on, and F2 generation also to know what is going on. So we'll go to the next slide, so we'll be able to explain F1 and F2 generation. So monohybrid inheritance, as I've earlier said, is actually the first law of inheritance, first law of inheritance. Now, first law of inheritance is actually Mendel's first law. Mendel's first law, which is law of segregation. Mendel's first law, which we call law of segregation. What does it say? It states that characters are controlled by it. Okay, so okay, good. I think I'm back. So like I said, Mendel's first law, that's first law of inheritance. It is also called the law of segregation. So they can say the law of segregation, Mendel's first law, or first law of inheritance. Now, what does it say? It says that characters are controlled. Now, for example, characters is your skin color. So your skin color, which is a character, is controlled by a single pair of genes, meaning it's either you are yellow, you are light in complexion, or you are dark in complexion. Although we have different skin color, a lot of them based on different locations. So, but let's look at the color of this very plant that he talked about now. Red, white. So this character is controlled by a single pair of factor, which is called gene. Single pair of gene, that is what control it, meaning gene, 2 pair of gene, and they will always be an allele. If you remember the definition of allele, one will be talking more about the other. So which at gamete formation, that is during gamete formation, though, this single pair of gene we now separate into different gametes. I'm going to explain that from the very crossing that we'll be doing now. So Mendel's fourth law, it is called the law of segregation. 1, in relation to a single pair of gene, contrasting characters segregate in F1. Contrasting character segregates in F1. Now, since I have it here, I think it's very easy for me to explain it here. I would have go and make use of a white what's it called, a whiteboard, but I don't think we really need it here because it is self-explanatory from what we have here. Now, the first law says, remember, I just want to use the law to explain this crossing. It states that characters are controlled. Now, what are the characters we have? Characters is a color. We're talking about color. So capital letter R for red color, small letter R for white color, a white flower. This will be like the key we are using. And it is very important during the examination period, because if your examiner doesn't see the key you are using, they might decide that they don't really understand what you are doing. That is wickedness, actually. But yes, they are right, because they don't understand what you are saying. They will say they don't understand. That is just it. So you must make sure you have the key. The key is actually what is telling you about the character. So we have the character, which is color. So we have red color, capital R, white color, small letter R. We now said they are controlled by a single pair of gene. Now, where's the pair? What we are now the phenotype, the physical look. What are you seeing? You are seeing a pure line red flower. You now want to cross it with a pure line white flower. What does that mean? A red flower that is totally red is not mixed with anything. It is not heterozygous. It is purely homozygous. You are crossing it with what? A pure white flower. Now, we said it is controlled by a pair of gene. This is now the pair of gene, parental genotype. This is the gene, RR. These are pair. These are pair. RR, pair. This is another word, RR, which is what? Pair. Now, we now say this pair of gene, during gamete formation, they will do what? They will separate into different gametes. And that is what we have here. Gamete 1, circle it. It's not pure circle, it is half circle. Gamete, other one, half, half, half. What is this telling you? This is telling you that this is one sperm cell, this is another sperm cell. This is one egg cell, this is another egg cell. Because you will assume one is male, the other one is what? It's female. So you do your crossing. Capital R to small r, that's 1 to 1, 1 to 2, 2 to 1, 2 to 2. So all that you will have here, they will be F1 genotype. Take note of genotype. Genotype is the what? Is the genetic constituent, and it will be represented by what? These codes. Now, the F1 phenotype, what will be the physical appearance? The physical appearance is they will all be red flower. Remember, Mendel's law talked about complete dominance. And for any complete dominance, it means a dominant gene will dominate the recessive gene. So here now, the red flower has a dominant gene. The white flower has what? A recessive gene. Quite easily done. So what will you be seeing? The phenotype you'll be seeing is all red flower. But the red flower is not homozygous red flower. It is what? Heterozygous, because it is mixed with what? White. But in F1 generation, you will not even see the white at all. All the offsprings, all the new flowers that will come out from the crossing, you will not see white at all. Everything will be red. And you'll not be feeling like, where are all the whites here? And the same thing happened to Mendel too. He's like, where are now the whites here? I mix white with this, and where are they? That is complete dominance. Now, what did he now do? He now picked the offspring, the flower. The flower now developed to form a fruit, and from there a seed. He now picked the seeds. Are you getting me? He now picked the seeds now and made 2 of the seeds. Are you getting me? He picked from this one, he picked from that one. So that is what we are seeing now. So he now made it for F2 generation. Okay, I'll take you there later. So he now mix them together to give the F2 generation. Now, what happens in F2 generation? This is the offspring from F1, 1 offspring, 2 offspring. We just pick 2 out of it and we move it to the next stage. So what will you be seeing? What is the phenotype you are seeing? The phenotype you are seeing is pure red. Is red. It's not pure line now because it is heterozygous red. So the phenotype is still red flower crossed with what? Red flower. What will be the genotype? Capital R, small r, capital R, small r. At gamete formation, what happened? They separate into gametes. You now cross everything the same way we cross above. What will you notice? You now notice that capital letter R goes with, that's 1:1, capital R, capital R. capital R, small r, capital R, and small r, small r. So what did you notice? At the F2 genotype, we have a pure line red. That's one pure line red. We have two heterozygous red, which is still red too, phenotypically. And we have one white. So what ratio do you notice? What is the phenotypic ratio, phenotype, the physical appearance? You'll be seeing 3 red. So what you'll be seeing here is 3 red, 1 white. That's 3:1. So what is the genotypic ratio? The genotypic ratio will be what? 1:2:1. What do we mean? 1. This and this one, they look alike. That is 2. And this will be 1. So 1:2 what? Ratio 1. So what are the genotypic groups? Because they do ask that question. What are the genotypic groups? What are the groups? This the group of new offspring that you can see. You can see 3 new group, the 2 capital R group, 1 capital R, 1 small r group, and the 2 small r group. So that will be 3 group, which is R, capital R, small r, and small r, small r. So that explains what Mendel's first law is talking about, which is also called the law of segregation or first law of inheritance. All you are saying is it states that the characters are controlled by just one pair of genes, and this pair of genes will separate during what? Gamete formation. From your crosses, you can give the whole explanation to what you have here. So we go to the next one, which is more like the summary. So the whole summary of what we did there is what we have here. And I think I've explained it very well, which I believe you should understand that. So in F2 generation, what do we notice? 3:1 for phenotype or phenotypic ratio. And the genotypic ratio is what? 1:2 what? 1. So just go through it. It's direct. It's one of those things we've talked about. Okay, let me go through it so that you'll be able to understand some stuff. So regarding the transfer of a single characteristics by each gamete and the resulting genotype can be demonstrated by mathematical probability. This probability of a gamete cell from a heterozygous F1 parent containing either the dominant allele R or the recessive allele small r is 50% or 1/2. So what are we trying to say? Remember, we only pick 2 offspring from F1 generation. So which means what we actually use there is what? Half of it. And also, if you look at what we pick capital letter R and small letter r, which is heterozygous. What does it mean? It means capital letter R is 50%, small letter r is what? 50%. Do you understand now? So if they now say what is the ratio of capital R to the ratio of small letter r, it is what? 50 out of 100 or 1/2 ratio. As simple as that. Now, if each gamete is represented by half, what do we mean? Capital letter R is half, small letter r is what? Half. If the 2 of them are represented by what? Half, the number of possible combination of F2 genotype is represented by what? Half times half, which is what? 1/4. What are we trying to say? We are talking about what is the probability? What combination can we see when half of capital R and half of small letter r, when they now cross? So what we are trying to say is we are going to see 1:4, 1 out of every 4. And that is exactly what happened during this process. 1 out of every 4. You will see this 1 out of every 4. So that is the ratio. So if each gamete is represented as by 1/4, the number of possible combination of F2 genotype now will be what? Half times half. 1 out of every 4. That will be it. So if there's another 4 again, there will be 1. And that is how it is. Hence, there are 4 possible F2 generation. 4 possible F2 generation. So somehow we've explained it in the previous one. So just go through it and you'll get more understanding. So the next one is we are moving into dihybrid inheritance. This dihybrid inheritance, I want you to listen very well because there are a lot of little things that are there. And I want you to understand it very, very well. If you understand it, that is just it. You've gotten it. As simple as that. In every examination or wherever you see it, you can do it without stress. Now, dihybrid. Di means two. Now, the first one, which is monohybrid, Gregor Mendel actually considered only one character. Here now, you now want to consider two characteristics together at the same time. For example, he wants to consider color of a seed and the shape of that seed. Color of that seed and the shape of that seed together, the hybrid inheritance. So it is the inheritance of two pairs of contrasting character. I think that is explanatory from what we explained. Mendel, in his investigation, by crossing a pure line round yellow seed with a pure line wrinkled green seed, obtained in line F1, which were all round yellow seeds, as usual. Remember, for every F1 generation, you will always see the same thing, meaning everything will just mix up, and that is just it. But before we go too far into that, let's just go to the crossing. So I know the crossing will be able to explain a lot of this. And once you understand it, that is just the end. Now, this is the crossing here. Remember the key. You always remember the key. Now, he picked a seed. He picked a seed. And he now look at the seed and notice that, okay, a round seed has yellow seed. The shape now, round seed, always have color yellow seed. And you now notice that the one that are wrinkled, they are not round, wrinkled, different, they have green seed. So which means these are two characteristics for two different plants, two different species, but the same plant, which is still pea plant. Now, he wants to do the F1 generation, he wants to do the crossing of the F1 generation. The first thing is this. What is the parental phenotype? Round yellow seed. We are crossing it with what? Wrinkle green seed, right? So what will be the genotype? So the genotype will be round. Remember, every character is represented by what? A pair of genes. So what are the keys we want to use? Let capital letter R represent round. Let capital letter Y represent yellow. Let small letter R represent wrinkle, and let small letter Y represent green seed. I think I've done that in the previous page, and this is just what I'm trying to say. Capital letter R for round seed, capital letter Y for yellow seed, small letter R for wrinkle seed, and small letter Y for green seed. And we now decide to explain it that the capital R is dominant, the capital Y is dominant, but the small r is recessive, and the green color also is what? Recessive. So that is to tell you the one that is dominating over the other. Always remember, Mendel's law is also called complete dominance. There will always be complete dominance, meaning one will always completely dominate the other. So we have the key here, and now we want to go to the crosses. Now, let's go there. Now, this is the first plant here. This first plant has round and yellow seed. So we are going to first cross round and yellow seed together. Why are we crossing it? It's more like you have bread separately, you have butter separately. You first want to mix bread and butter first. You do it for the first plant, for the first seed, and you do it for the second seed. Then the gene would have mixed together. Why are we doing that? We are just trying to say that the genotype, we need to undergo meiosis to form gametes. And when they form gametes, there will be what? Crossing over and all those stuffs, and later form a new species, which will now become 2n number, which become n number of chromosomes, which is haploid number of chromosomes, before it will now mix with another plant. That's what we are trying to explain here. So capital letter R, two capital R here, two capital Y. So at gamete formation, they separate R, cross with Y, Y. So we cross it RY, 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 RY. So the gametes you will have now, all the gametes you have will be RY, RY gamete, RY gamete, RY gamete, capital R. And for the other one too, small r, small y, you do the same thing too. You have small r, small y, small r, small y, small r, small y. What does that mean? It's telling you now that what? These are in their what? In their gamete stage, meaning the wrinkle and the green color, they are together. Are you getting me? They'll be inside the same sperm cell. The sperm cell will now take it out and go and meet another egg cell of this very other one. So that is exactly what is happening here. Now, take two gametes each from the gametes above and cross to get the F1 genotype. You take two gametes, 1, 2, because these two gametes now, they are like the sperm cell 1, sperm cell 2, egg cell 1, egg cell 2. So you move it down. R, as you can see here, I've moved it. Now cross with what? The smaller one, which would be the parental genotype. So at gamete formation, each of them will now what? Go into the same circle again. If you remember, this capital R, this is small y. Sorry. I think somehow it got erased. So what do? You now cross them. If you are not crossing them, you will make sure you bring those ones that have the same alphabet together. Not because you say R and Y, RY, small r, RY. No. You make sure you bring the alphabet together so everyone will be together. So for the F1 generation now, for the F1 genotype, you have capital R, capital Y going towards small r, small y. Then you have capital R, capital Y, small y. The same thing for the second one, the same thing for the third one, same thing for the fourth one. And you always remember, Mendel will always tell you that you are crossing pure line, and at the end of the whole thing, you will always have the F1 generation, which will always be what? Heterozygous condition, and everything will be what? The same thing. So from what you are looking at here now, remember, capital R is dominant over small r. Capital Y is dominant over small y. So what is that telling you? All you'll be seeing here is round yellow. Sorry, round round yellow, round yellow, round yellow, round yellow. That is all you'll be seeing here, because the round and the yellow have completely dominated the wrinkled and the green seed. So in F1 generation, you will always have the dominant gene expressing themselves. I think that is understandable. Now, we now go to the next one now. You will now take two, remember, how do we get F2 generation? F2 generation is gotten from what? The crossing between two F1 generation. The crossing between two F1 generation will always give you your F2 generation. So exactly what we are going to do now. So F1 genotype, all will be what? Capital R, small r, capital Y, small y. F1 phenotypes, all heterozygous round yellow, which I just finished explaining. Heterozygous round yellow. As simple as that. Now, we now pick 2 out of the F1 generation now. This is 1, 2 out of the F1 generation. That's what we move here. Round and yellow seed, round and yellow seed. But remember, that is the phenotype. So the genotype will be what? Heterozygous. Capital R, small r. Capital Y, small y. Are you getting me? So, which means this is the genotype. At gamete formation, what happened? They will now cross each other again. They will cross each other. Now, when they cross each other, what do you see? Capital R come down, small r come down. Capital Y come down, small y come down. These are the gametes. You now cross them again. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. As simple as that. Now, you do the same thing to the other side, but you will know they will be the same. Why would they be the same? Because it is the same genotype that you pick year and year, but you must do this. You must show the two so that whoever is marking will give you your full mark. You must show the two. Now, arrange the two results on both vertical and horizontal sides of the table below using what? Punnett square. Now, we now want to use Punnett square from here now. What are we trying to say? At the vertical side, you put capital R, small y, capital R. Okay, let me just show you straightforward. So we go to the next slide and we are going to arrange it. Capital R, small, capital R, capital Y. I hope you still remember what we have at the back. Capital R, capital Y, capital R, small r, capital Y, capital R, small y. I hope Y, R, capital Y, R, small y. Okay. Small r, Y, small r, big Y, small r, big Y, and finally, small r, small y, small r, small y. Right. That is where the mistake has been coming from. Okay. So to correct the whole thing now, this is where the problem is year and year. So this very man here is going to be small r. This very man here is going to be small r. This very man here is going to be small r. And this very man here, the two of them here will be too small r. Hi. Sorry. That's, I think, moving the whole thing caused yeah, maybe I plug what's it called? I've clicked on the cap lock. So change the whole thing there. So that's why I said you have to be very careful. Even myself now, for you to see that I needed to go back and check where the mistake is. So I think from here now it makes everything um kind of easier for us. I think we still have this correctly. The capital R, small r, and two capital Y, the two yellows, they are still correct. The blue is still correct. This very one might not be correct again. Yes, because something has changed here. Something has changed here. Something has changed here. Okay. So something has changed here. That is 3. Okay. Let me see. Capital R, small r. Capital R, small r. Capital Y, small y. Okay. This also. Okay. I think I need to rectify a lot of things. Capital R, small r. Capital Y, small y. Okay. Capital R, small r. Small y, small y. Okay. Small r, small r. Small r, small r. Okay, good. So this, that is 3. Is there another one like that here? This will be 3. Capital R, small, capital Y, small, wrong. And okay, I think we're on track now. So the next one, 2 capital R, 2 small y. 2 capital R, that will just be 1. 2 capital R, 1 y. That will just be 1. So those ones have changed now due to the mistake I made here. Sorry for that. I think I make this class a bit longer now because of this mistake. Now, we have the capital R. Okay, this is already here. So the next one we want to look at is capital R, small r, 2 small y. 2 small y. Capital R, small r, 2 small y. This is 1. This is 2. This is 1 and 2. 2. I think it's just that 2. Okay, good. 2 small r, 2 small r, 2 big y. Okay, that is 1. 2 small r. Okay, let me give it a color too, so that everything will be clear to us. Now, this one is 2 small r, 2 small r, 1 capital Y, 1 small y. This is 2. Very good. This is 1. And this is what? 2. And the last one is 2 small r, 2 small y. Okay, that is what? 1. I think there's one that I omitted again. I'm not sure. I'll check it through now so I will know what is going on. Okay, there's one I did not paint here, so I'm not sure if we use it or not. 2 small r, 1 capital Y, and 1 small y. So it looks like this green. 2 small, 1 capital, and there's 3. Wow. 2, 3. Green. Green. I just hope we've been able to rectify this mistake. Okay. So the round yellow, 3, 4, 5, 6, 7, 8. And that's still here. 3, 4, 5, 6, 7, 8. Supposed to be 9. Round yellow. Round yellow. It's supposed to be round yellow too. Hi. Something has happened here. 2 small r, y, and y. Aha. Yes, this is exactly what happened. It is the problem is here. The problem is here. And why is it here? Small r, small y, capital R, capital Y. So this is supposed to be capital R, capital R, small r, and capital Y, small r. So this is going to go with which ones here? This, which is the pink gang. The pink gang. What's going on here? Go with the pink gang. Pink gang. So this is going to be 4 here. Okay, that has been corrected. So 4, 5, 6, 7, 8, 9. That is the first 9. The second one will be 3. Okay, I need to remove this. I need to adjust this because I've adjusted it there. So this will be 9. Round green. This is 3. Wrinkle yellow, 2 plus 1, 3, and 1. Yes, correct. I think we've been able to correct it here now. So I would let me expand it again so you'll be able to see it clearly. So please, I think with this color, it will be very easy for you to put out what really happened. And okay, let me do it this way. Since we've taken our time to adjust everything, let me put all the colors that I use for each of them here, so that it will be easy for you to get it, especially when you are trying it yourself. Capital R, do this, that is yellow. And this very part here, capital R, small r, capital R, small r, and 2 small y. I used the ash color. Okay. And 2 small r, 2 small r. Okay, I use blue. I use this blue. Okay. Small r, capital, and this. Okay, I use the deep green. Yeah. I use the deep green. I think so. No, I use the deep green, not this. Beautiful. And the last one, I use this. What did I use? Too small. Okay, good. I think I used this. Okay, beautiful. So now you see that this actually wasted a lot of our time. I think the first mistake came um as a result of this. Instead of small and small, as you can see now, the tally with what we have there: capital small, capital small, capital small, capital small, small big, small big, small small, small small. So I think the whole mistake came from that very part, because somehow I use capital R for this. So at the end of the whole thing, when you calculate it, you must have 9331. That is 9 round yellow. These are the round yellow, round yellow, round yellow, round yellow. 4, 5, 6, 7, 8, 9. That's 9. Round green. How many round green? 2 plus 1, 3. How many wrinkled yellow? 2 plus 1, 3. And the last one, round green, which is 1. So Mendel's second law is called the law of independent assortment of characters. Now, characters, remember, this here now is 2 character, not like the first one. So first thing you have to do is you have to independently sort them out and sort them, because we have 2 characteristics there. Now, in relation to 2 or more pair of gene, segregation occurs independently. Because we are talking about 2 or more gene, there must first be kind of segregation. And this segregation, which normally every gene will undergo segregation, but this segregation here now must be independently, meaning they must not depend on each other. You, color, do your own separately. You, shape, do your own separately. It's as simple as that. Now, let's quickly look at this table. So this table shows us the difference between monohybrid and dihybrid inheritance. The phenotype group in monohybrid is just 2. If you go back to it, in dihybrid is what? It's 4. Genotype group 3. Here it is what? 9. If you count all this, 1, 2, 3, 4, 5, 6, 7, 8, 9. If I'm right, yeah. 1, 2, 3, 4, 5, 6, 7, 8, 9. That is for the hybrid inheritance, which is second law. But for the first one, it's just 3. Number of gametes, we have 2 gametes. In the other one, that's in monohybrid, 2 gametes. But here, 4 gametes. Why is it 2 gametes? Because in the first one, it is just monohybrid inheritance. Monohybrid, 1 character. That's what you are considering. So there will be 1 for the for tall, 1 for short. But in dihybrid, 1 for tall, 1 for short, 1 for red, 1 for yellow. So 4. Number of offspring in monohybrid, you have 4 offspring. Here, you have 16 offsprings. If you count 1, 2, 3, 4. Okay, let me come back here. Aha, this is it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. All these are the offsprings. So 16 offsprings. What is the phenotypic ratio of monohybrid inheritance? Just 3:1. But in dihybrid inheritance, that is what? 9:3:1. 9:3:1 phenotypic ratio, dihybrid inheritance. Now, what is the genotypic ratio? If you look at the genotypic ratio, 1:2:1 for this. But for this, 1:2, see how long it's going to be, as long as anything there. So that is that. Now we go to the next slide here. We've actually wasted a lot of time on that. And I don't want this slide to be, this class will extend more than 4 classes. And this is our 3rd class. But I think it's better you understand it. And the way where we made a mistake, it make me begin to use color and everything to differentiate it. And I think with that, it will make a lot of you quickly get it properly compared to the way I was explaining it before. So, question now. We've talked about monohybrid inheritance, dihybrid inheritance, and from that very 2 part alone, IJB and JPEG have a lot of questions to ask you. So let's look at an example here now. In Snapdragon, red flower color R. Now they've told you that the red flower color is represented by what? An allele, which is what? R. Is incompletely dominant. Now, this is one of those things I'm talking about. Remember, under Mendelian principle, it is complete dominance, not incomplete dominance. Now, this incomplete dominance is a deviation from Mendelian principle. It's a deviation. We'll be discussing it after this question, but I decided to bring in this question so that you will know how to make use of that Punnett square. And especially, I want you to practice it on your own. In fact, it is just the same thing as you are copying and pasting what I just finished explaining to you now. You are just copying and pasting it. But the only difference now is there is incomplete dominance, which means when red mixes with white, it will change to what? Pink, depending on what they ask you to, it will change to. But let's read this question. So this question will tell you now. Is incompletely dominant to white the heterozygous condition, meaning when you have capital letter R and small letter R now. Small letter R will be for white. So when you have capital letter R and small letter R now, automatically, that is heterozygous condition. You now said it is going to give you what? A pink flower. Now, the heterozygous condition now has given you a new color. It is no more red again. No. It is now a new color. So which means after crossing over, you will now be seeing red, white, and pink color. And broadleaf capital letter B. Now, okay, that is the first one. Now, they first explain the first one. They said red flower incompletely dominates white flower. It will now give you another new species, which is what? A pink flower. They now said also looking at the leaf. So here now they are considering flower color and leaf shape and leaf shape now. And broadleaf capital letter B is incompletely dominant to narrow leaf small letter b. They now say the heterozygous condition, when you have capital letter B and small letter b together, which is heterozygous condition, it will now give you intermediate breadth. That is the new name. Intermediate what? Breadth. So you are not going to say capital letter B completely dominate small letter b. No, the name has changed to intermediate what? Breadth. They now said if a red flowered, broad leaf plant, that is capital R, capital B, capital B. You remember all those now. That's where we are coming from beginning of what we did. We just finished now. So, okay, maybe I can use white pen to just explain that too, so you'll be able to understand it. I'll just do it to a level. So from there, you can continue. So they said, if a red flowered broadleaf plant is crossed with a white flower narrow-leaved one, what will be the F1 and F2 generation? Remember how we get the F1 generation, and from the F1 generation we took 2 offspring and take it to what? F2 to give our F2 generation. So they now said what will be the F1 and the F2 phenotype? Using diagram to show how you arrive at your answer. But remember here now, something will change. Something will what? Change. What will change? All this heterozygous condition here, all this heterozygous condition here, it will change. It will change name. It will not be round. No, it will not. Everything will change. So this is what I will do for you. This is what I will do for you. How should I explain it now? It's not going to take our time. Okay, let's do it. R and B. R and B. Let me quickly use a whiteboard. Let me use a whiteboard so that we can do something from there. Okay, this is a whiteboard here, and I'll be using my hand to write so that I'll be able to get something. The only issue I will just have now is I'll have to be going back and forth to get that information. So red, if you remember, they said capital letter R, which is red. That's R. They said the other one is white, if I'm correct, which will be small letter R. And they said the other one is broad, capital letter B. Broad, capital letter B. And I think the other one is narrow. I'm not very sure of that, which will be small letter B. And they now said the heterozygous condition, that is when you now have capital letter R and small letter R, it will not be the heterozygous condition will now give you what? Thing. Name has changed. This one too, the heterozygous condition, capital B and small b, will now give you what? Intermediate. No worries, my hand I'm using to write on my laptop. My laptop is a touch screen. So I'm using my hand to write. So it might not be very clear, but I believe you should be able to see it clearly. So that is, I just summarize everything that we have there. So what are you now expected to do? Remember, if you want to do anything, let me see. Let me move it to one side. I just hope you can see clearly what I am doing. Okay. I hope you can see. Now, if you want to do the crossing now, what are we going to do? No. Okay. I want to move. Oh no. I want to move it to one side. Beautiful. So now let's see how the crossing will look like. I still want this to be here so that you'll be able to see it. So for the first one, capital, the genotype will be 2 capital letter R with what? Broad leaf, which is capital 2 capital letter B. We are going to cross it with 2 small letter R and 2 small letter B. But at first, what will happen? You will make sure each of them first go into gamete formation, which will be R, R, cross with B and B. And you crop R capital B. In short, everything will be R capital B, R capital B, and R capital B. Make sure you do your correct crossing though. I'm just doing this because I'm using this whiteboard. Okay, I've corrected it. So here now will be what? Capital small R, right? Small R cross with small b and small b. So you have RB, you have RB, you have RB. Sorry, this is small letter arrow. This is one of the mistakes we do talk about. And small r. Very good. Now, after we've done this, if you remember, I hope you are looking at it here. Just look at it because, like I told you, I'm using screen, so which means I might need to, I might need to move it to another side so that we can be able to use the other side too. So since we've gotten this now, remember, we'll now take 2 of this offspring, 1, 2, and cross it with what? 1 and 2. So we have RB, circle, RB, circle, cross with RB, circle, RB, circle. This is where I'm going to stop. Once I get the F1 generation, I'm going to stop so that you not continue from there. All I just want to establish, I want to establish is the what's it called? The intermediate when it changes color. When the color changes and everything. That's what I want to establish. So here now we have capital letter R, small letter R, capital letter B, and small letter b. Remember, everything will be the same thing. Remember, everything will be the same thing. Everything will be the same thing. And that shows that what? It is F1 generation. All F1 generation. This is it. So what do you notice here? At F1 generation, all will be intermediate. And they've already told us in the question that all the intermediate, when you see capital R, small r, it will be what? Pink. When you see capital B, small b, it is what? Intermediate. So that will be what? Pink intermediate. This will be what? Pink intermediate. That is the phenotype. Pink intermediate, pink intermediate, pink intermediate. That's how everything will be. Now, the pink intermediate now, you will now take two offspring from F1 generation. You see that you've answered one question already. So all this will be what? Four pink intermediate phenotypes. The phenotype will be what? Pink intermediate. You see that it has changed totally. We don't talk about red again. We don't talk about broadleaf again. Everything has changed name. That is what we call incomplete dominance. Now, from here now, you now remember that it is from here now that we now take two of this and cross it again. So that will make you go back to what we now did. You now cross this again, capital B cross small b. I hope you are seeing what I am doing properly. So when you cross it, you have what? Capital R, capital B, capital R, small b, small r, capital B, and small r, small b. So this is what you will now do in two different places. You will now move it to the Punnett square. You will now move it to the Punnett square. So this will be RB, R small b, R capital B. Don't let me make mistake again. Capital R small b, small r capital B, and small r small b. As you can see, then you now begin your Punnett square again. R small r capital B and small r small b. So you now start your Punnett square all over again. You now begin to do it. Now, you will now notice that when you are now plotting that your table, so I think with this now, I think you understand what you are going to do. So let me go back to um let me go back to our explanation. Okay, very good. So by the time you now cross it, you will now get back to this place. You know, we've gotten here already. So you now cross everything. So what will now be the difference now is all this one that are showing you, okay, like this one now, they are capital, 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 capital. So this one will still remain red broad, red broadleaf. So this intermediate will now be pink, pink broadleaf. This one will be red intermediate. This one will be pink inter pink intermediate. Yes, pink intermediate. That will not be the difference. So at the end of the day, the phenotypic ratio will not be 9331. That is exactly what they want to know. That is why they are asking the question. The phenotypic ratio will be equal to the genotypic ratio, because everything will be different from one another. And that is to show you that it is a deviation from Mendelian principle. Simple as that. As simple as that. As simple as that. Now, what I'm going to do is this. At the end of this class, I would, what did I want to do? What can I do? I want to see everybody. I want everybody to do it. I want to see, I want to see your answers so that I'll be sure that you've gotten it. If I give an email address, a lot of you will drop it in the email. Or basically, don't worry, just submit it to your coordinator. I will communicate every coordinator to send it to me. Each center to send it to me. I think from there, it will be easy for me to assess it and see which center actually understand it and which center do not understand it, so we can continue from there. So that is all about the Punnett square. Automatically, we've answered question 1. Now, the question they asked, if you remember very well, they said, what will be the F1 and F2 phenotypes? Why do they ask that question? Because they know you are familiar with 9331 of Mendelian principle, which is for complete dominance. They now ask this question. They make sure it is what's it called? It goes alongside with Mendelian principle, so that you will mistakenly say that the phenotype is 9331, which is wrong. And the reason why it's wrong is because it is not going to follow the Mendelian principle, because it is a deviation from Mendelian principle, because the intermediate or the heterozygous condition has another new name. It's just as simple as that. So the second question here, the heterozygous couple whose, oh, sorry, a heterozygous couple whose genotype is capital A small a have an albino child whose genotype is AA. So diagrammatically, the probability of their next child being A, an albino, B, a carrier, C, a normal. Now, what they are trying to say here is what? Heterozygous couple, which means both of them has capital A, small a, heterozygous. Since they are couple, the two of them has this. So if it is individual, they say the father has this, the mother has this. But they said a heterozygous couple has genotype, whose genotype is capital A, small a. What are we trying to say? They have heterozygous condition. And because it is an heterozygous condition, the two of them are not albino. But because of that recessive gene A, that the two of them carry, remember, it is not a sex-linked character. Yeah, it is not a sex-linked character. If it is a sex-linked character, the man will have it. Like the man will be an albino. But because it's not a sex-linked character here, so it's just carried by one of the gene. So that is why we are representing it by what? Capital A and small a. If it is a sex-linked character, we are going to put X raised to power A or raised to power whatever it is. Now, let's go to the next slide so that you will see the solution to that. So the parents are capital A small a crossed with capital A small a. This is the father, this is the mother. When you cross it, you have AA, capital A, capital A, capital A small a, capital A small a, and AA. So the question says, what is the probability of their next child being A, an albino, B, a carrier, C, a normal? So the probability of their first child being an albino is 1/4. That is 1 chance out of 4 children. I think that is understandable. One child will be an albino out of 4 children. That is 1/4. Probability of being carrier. Probability of being carrier. Now, how many are carriers here? A, 2 of them. So carriers will now be what? 2 out of how many? Out of 4, which is the same thing as what? 1/2. Probability of being genotypically normal. Probability of being phenotypically normal is, who is normal here? Only one. Number one is normal. One out of what? 1, 2, 3, 4. That is just the answer to the question. As simple as that. And the last question here, they said in pea plants, round seed shaped R is usually expressed. Why wrinkle? Remember, capital letter R is expressed, which means it's dominant. Why wrinkle seed shaped small letter R is suppressed, which is the recessive gene. They now said a cross is made between a male heterozygous, round seeded plant, and the female plant breeding true for wrinkled seeds. What will be the appearance of the seed of F1 generation? I don't know if you are following it there, but fourthly, they've shown us what the answer is going to be. In pea plants, round seed shape capital R. That's round seed carrying capital R. It is dominant, express. The wrinkle seed small letter R is suppressed. The now said a cross is made between a male. The male is heterozygous round. What does that mean? Capital letter R, small letter R. Right? And the female plant breeding true for wrinkle seed. When they say something is breeding true for wrinkle seed, it means it is too small letter R. What will be the appearance? Now, this very question, I'm expecting you to know where they are going to. Because under test cross and back cross, I explained something like this to you. This is more like the question under back cross, test cross. Back cross, test cross. Now, in short, they said the male is what? Heterozygous round seed, heterozygous, capital R, small r. The other one is breeding true for what? Wrinkle seed, which is too small letter R. The gametes, you cross it. Capital R, small r, capital R, small r, small r, small r, small r, small r. So in F1 generation, what are you going to have? You have two round seed, which is 50%, two wrinkle seed, which is also what? 50%. So it will be 50-50. 2A. Now A, sorry. The other one now says selfing the offspring of this parental cross. Okay, I think that's what we did first. And the offspring of the cross between the male and, okay, let's see this. So selfing now, what does it mean to self? Self means you now take the two offspring from the first one out, two offspring from the second one. Definitely, you know where the two offspring from the second one will be. You know what it's going to be because everything will be more letter R wrinkled. But selfing the first one, which is the two heterozygous, take two offspring from one, you are selfing it. Selfing means those ones that look alike, you bring them together and you do a cross on them. So capital R, small r, what are you going to have? Two R, heterozygous one, and one wrinkle. Okay. So let me see if I didn't miss anything out that I want to explain there. Oh, window is closed. Oh, sorry. I think I'm almost rounding up the time here. I'm using more than the time given to me. I think I'm using more than the time given to me. So I might decide to end this class soonest. So yeah, that is just it. Round, remember, and wrinkle, heterozygous, homozygous, and this. And finally, 2B. 2B says, okay, the other one, remember, they said you should self the two, selfing. So I've told you already before that this wrinkle, everything will all be what? Wrinkle. Now the next question now says, The offspring of a cross between a male heterozygous round, cross between a male heterozygous round of the F1 generation and a female homozygous round seeded plant. Homozygous round seeded plant. So that is heterozygous round seed and homozygous round seed. So you cross that also. What are you going to have? All are going to be what? Round. You have two homozygous, you have two heterozygous. Now the fourth one now, see what are the phenotypic ratio of everything from beginning to the end. So you have round ratio wrinkle, round ratio wrinkle, round ratio wrinkle, 2:2, 1 ratio. Sorry, I don't want to I don't want to go back and so what are the genotypic ratio in the progeny of the plant named above? So all the crosses that we did here from here downward is what we now give the genotypic ratio. You understand that 2:2, 1:2, 2:1, 0:4. 0 means there is the other one is not there, but one is there, the other one is not there. So 0 means it's not there at all. 4 means we have 4 of it. So that is how we now give the genotypic ratio of everything here. Now, as I've been talking about, this is now where we are starting the deviation from Mendelian principle. And there are three main deviation from Mendelian principle, which we've used time to solve question on one of them, which is incomplete dominance. We have three deviation from Mendelian principle. 1, incomplete dominance. 2, codominance. And 3, linkage. Linkage. So I don't know. I think I want to end this class here so that it will not be too much for you guys. I will now continue from this deviation in our next class. So that it will not be too, because I think we solve a lot of questions here. So I want you to have time to go through it and understand it better against the next class. I don't want to rush you at all. So the next class now, I'll continue from here and we can go through the whole thing one after the other. So thank you very much and remain blessed. Remain blessed. to everyone. So I'm very sorry. It's going to be so wordy